Part 2 of the study guide. Structural diversity reflects functional diversity in globular proteins. In globular proteins, the polypeptide chain or chains folds back on itself to form a more compact structure. Myoglobin provided early clues about the complexity of globular protein structure. Myoglobin was one of the first globular proteins to be studied. It binds O2 in muscle cells and facilitates O2 diffusion. The protein surrounding the heme of myoglobin consists of 8 right-handed alpha helices connected by bends, some of which are beta turns. Most of the hydrophobic R groups are in the interior, demonstrating the importance of the hydrophobic effect and the entropic effect. Weak interactions strengthen this interior core. All the peptide bonds are in the planar trans configuration. Fe2+, iron in its ferrous form, in the heme, has 2 perpendicular coordination bonds, one to the nitrogen of the proximal histidine, histidine 93, the other left open for O2 binding, but access to this coordination bond is restricted because otherwise Fe2+ can quickly be oxidized to Fe3+, which does not bind O2. Studying myoglobin helped researchers understand how structure dictates function. Globular proteins have a variety of tertiary structures. Globular proteins have a hydrophobic core, a hydrophilic exterior, and are stabilized by weak non-covalent interactions and entropy. Three-dimensional structure may be considered as an assemblage of polypeptide segments in the alpha helix and beta conformations. A motif or fold is a recognizable folding pattern involving 2 or more elements of secondary structure and the connections between them, and it is a kind of supersecondary structure. An example is a beta-alpha-beta loop, which is 2 beta sheets connected by an alpha helix. A motif may be more complex, such as a beta barrel, shown below, which is a barrel of interweaving beta sheets. Fold tends to refer to more complex patterns than motif, but they are used interchangeably. A coiled coil is a motif, and the 8 alpha helices of myoglobin is a motif. A domain is another term describing a structural pattern, specifically a part of a polypeptide chain that is independently stable or could undergo movements as a single entity with respect to the entire protein. In a protein with multiple domains, each domain may appear as a distinct globular lobe, but typically domains are hard to distinguish from one another due to extensive interactions. 4 common folding patterns. 1. Hydrophobic effect influences stability and the entropic effect. 2. Alpha helices and beta sheets are typically not adjacent because they cannot hydrogen bond to each other. 3. Segments next to each other in primary structure typically are next to each other in folded structure. 4. The beta conformation is most stable when the individual segments twist slightly right-handed, and 2 parallel beta strands tend to be right-handed due to it being more compact. An alpha-beta barrel is a beta barrel where each parallel beta segment is connected by an alpha helix, and all connections are right-handed. These are often found in enzymes with a binding site at one end of the barrel. Some proteins or protein segments are intrinsically disordered. Some proteins or protein segments lack an ordered structure. Around 30% of human proteins may be intrinsically disordered, having no set structure but clearly defined functions. Proline residues are common because they tend to disrupt ordered structures. IDP have high densities of charged amino acid residues. IDP may serve many different and important functions, such as wrapping around other proteins and preventing them from working, which can be useful. Protein motifs are the basis for protein structural classification. There are many protein databases. Most proteins have similar folding patterns. A superfamily is 2 or more families of proteins that have little similarity in primary structure, but considerable similarity in secondary and above structure, and in their functions. Some proteins act similarly, therefore evolution is highly probable. Protein databases are important. Protein quaternary structure range from simple dimers to large complexes. Proteins can have multiple polypeptide subunits from 2 to hundreds, and these serve many roles. Sometimes subunits serve different functions, sometimes related. Multimers have multiple subunits, oligomers have few subunits. A protomer is a repeating structural unit in a multimeric protein, whether a single subunit or a group of subunits. Hemoglobin is an oligomeric protein with 2 alpha subunits and 2 beta subunits. The subunits pair as alpha-beta, so hemoglobin may be called an alpha-beta dimer, or a tetramer. Protein denaturation and folding. Proteostasis is the maintenance of a protein. Chaperones help proteins fold when they are synthesized on a ribosome. They may also help a temporarily unfolded protein to refold, which may help prevent misfolded proteins from sticking to each other in aggregates such as in Alzheimer's disease. Loss of protein structure results in loss of function. Denaturation is when a protein loses its three-dimensional structure, and therefore its function. Extreme heat may denature a protein through complex effects on the weak interactions. Extreme pH may denature a protein, typically through disruption of the hydrophobic core, or by disrupting hydrogen bonds, sometimes by changing the net charge, causing electrostatic repulsion and hydrogen bond disruption. Denaturation often leads to protein precipitation. Amino acid sequence determines tertiary structure. Renaturation is undenaturation, and it proves that tertiary structure is determined by primary structure. If a denatured protein of a certain amino acid sequence can be renatured, then tertiary structure depends on primary structure. Ribonuclease A can be denatured by a concentrated urea solution in the presence of a reducing agent, and renatured by removing the urea and the reducing agent. Very few proteins will fold spontaneously from primary structure to tertiary structure. Many require help. Polypeptides fold rapidly by a stepwise process. Proteins fold quickly. Leventhal's paradox concludes mathematically that a protein does not go through each possible folded conformation before reaching the correct one. Local secondary structures form first. Alpha helices and beta sheets may form. Ionic interactions between nearby groups may form. Next, longer range interactions occur, and eventually complete domains form and the protein is totally folded. Close range interactions tend to fold a protein faster. The hydrophobic effect and entropy play roles throughout. Unfolded conformations have higher conformational entropy and higher free energy. Semi-stable intermediates can slow the folding process. Thermodynamic stability is not uniform throughout a protein, and many people are working on predicting protein structure through programs. Some proteins undergo assisted folding. Two major families of chaperones are HSP70 proteins and chaperonins. HSP70 proteins are more abundant in cells stressed or shocked by heat shock proteins, and the 70 means that the molecular weight is about 70,000. HSP70 proteins bind to highly hydrophobic regions of unfolded polypeptides. Chaperonins are structured as a series of multi-subunit rings, forming 2 chambers back to back. The eukaryotic equivalent is HSP60. In HSP60, protrusions are at the apical domains at the apex, close over the chamber, trapping the target protein, preventing protein aggregation. Protein disulfide isomerase, PDI, catalyzes the interchange of disulfide bonds until the bonds of the native conformation are formed. Peptide prolyl cis transisomerase, PPI, catalyzes the interconversion of the cis and trans isomers of peptide bonds of proline residues. Defects in protein folding provide the molecular basis for a wide range of human genetic disorders. Misfolding happens. Some diseases are caused by misfolding, such as Alzheimer's, Parkinson's, and Huntington's, where a protein becomes an insoluble extracellular amyloid fiber. These diseases are called amyloidoses. Most proteins that assume an amyloid fibril structure have aromatic amino acid residues in a core region of a beta sheet or alpha helix. These aromatic structures interact, stabilizing the beta sheets next to each other in amyloid fibers. In eukaryotes, proteins destined for secretion undergo initial folding in the ER, but if unfolded proteins accumulate, the unfolded protein response, UPR, activates. Chaperone concentration may increase, and/or protein production may decrease. Amyloid aggregates that form before the UPR occurs may be degraded by autophagy, or by a system of proteases called the ubiquitin proteasome system. Some amyloidoses are systemic, involving many tissues. Some amyloidoses are associated with particular organs, such as in diabetes, where pancreatic cells cannot secrete insulin or regulate glucose due to the protein amylin, which accumulates around the cells. The amyloid beta peptide causes Alzheimer's. In Parkinson's disease, alpha-synuclein accumulates as Lewy bodies, which are spherical filamentous masses. Huntington's disease involves the protein huntingtin. All of the bad symptoms are highly suppressed if HSP70 chaperone expression is increased. Chapter 5. Oxygen combined to a heme prosthetic group. O2, molecular oxygen, cannot be carried to tissues if it is simply dissolved in blood serum. None of the amino acid side chains are suitable for the reversible binding of O2. Transition metals, especially Fe2+, help with O2 transport. Free iron cannot be used because it promotes the formation of highly reactive oxygen species that can damage DNA and other macromolecules. So Fe2+ is bound in a heme prosthetic group. Heme consists of protoporphyrin, which is the ironless organic ring, and Fe2+. Fe2+ has 6 coordination bonds, 4 to nitrogen atoms that are part of the porphyrin ring, and 2 perpendicular to the porphyrin ring. These coordination bonds help prevent conversion from Fe2+ to Fe3+. Fe3+ does not bind O2. Free heme molecules lack the 2 perpendicular coordination bonds, allowing O2 to react simultaneously with 2 free heme molecules, potentially changing Fe2+ to Fe3+ irreversibly. A heme surrounded by a protein, specifically the globular protein globin, thus restricts access to these 2 perpendicular bonds. One of the bonds is occupied by a nitrogen of the proximal histidine. The other binds to O2. Globins are a family of proteins with similar primary and tertiary structures. Myoglobin facilitates O2 diffusion in muscle tissue. Hemoglobin is important for O2 transport in the bloodstream. Neuroglobin is expressed largely in neurons and helps protect the brain from hypoxia or ischemia. Cytoglobin is found in the walls of blood vessels, regulating nitric oxide levels. Myoglobin has a single binding site for oxygen. Myoglobin, MB, consists of 8 alpha helical segments, A through H, connected by bends, AB through GH. The proximal histidine is called histidine F8, or histidine 93. And we'll pick up again later.